the last video we were discussing about pharmacological profiling. This pharmacological profiling of a new drug can be done by in vitro studies or by in vivo profiling. So already about the in vitro profiling we have discussed in the laboratory by using either cell lines or by using a tissue. The studies will be conducted outside the body. Today in this video let us see about the in vivo profiling. The in vivo profiling involves the testing of the drug on normal animal models. So we will be using various animals which includes rat, rabbits, mice for conducting the experiments. And coming across with the features of in vivo models. So these models are quite expensive and also they are time consuming. Very rapidly we are not getting the result. So then even these studies can be still extended towards the larger animal depending on the studies what is planned. So a particularly important role of in vivo experiment is to evaluate the effect of long-term drug administration on the intact organism. As in the in vitro studies, we will be studying the drug action, whether molecularly they are bounding with the target, so then whether they are having the effect or not. When coming with the in vivo profiling, where we will be using intact animal, like whole animals are used, so that what effect the drug can show in a system, because multiple organs are present in the body. Once the drug is administered, not only it will show its action on the desired side, but even though they are going to act on the other organ system also, and also there will be influence of some other organs on the target organ also. So how they are going to influence the drug action in detailing this in vivo profiling of the drugs, they are going to help us. This is the difference what you can come across with the in vitro models, because in vivo, so many of the organs are controlled by the central nervous system. In vivo profiling, so you will be identifying the role or the influence of central nervous system on various organ systems. But while doing the experimentation on the animals, many factors they are going to influence the drug responses. The first consideration what you should keep, that is the species differences, because it's important to take species differences into account at all the stages of the pharmacological profiling. Because if with one animal what the target you have taken, the similar response what you got with the selected target in the other species of animal may not be obtained, because there will be difference in the species. Because that's why we can conclude like the same target in different species will generally differ in their pharmacological specificity. So then how to overcome such aspects? So there are many possibilities, because rapidly there is an advancement in the animal experimentations also, because these animals as such healthier animals can be used, or one more option widely what we are using, that is we are manipulating the animals by giving some of the chemicals, so that we are bringing the symptoms of disease in the animal. So other possibility like what we have, that is the getting some transgenic animal itself. The advantages of transgenic animals, like so they will be in birth having the defects of the disease, so that they will show similar responses of a physiological disturbances in the human body, so that there won't be any influences. So that nowadays transgenic animal models are used of just by manipulating the gene, by in birth disease will be generated. So this use of this model with a human disease, for which the drug has been prepared, because with this what we are going to get is the answer for a crucial question, whether the physiological effect result in a therapeutical benefits or not. Many times some of the physiological responses what the drug can elicit may be because of the therapeutical benefit, or sometimes it may be non-benefit responses. If the animal models are prepared properly, so then exactly we can check whether the drug have shown the therapeutical benefit or not. So now The challenging factor is that despite we have a range of diversity of the animal models from humans, so there will be a valuable link which can be get generated because a lot of species differences we may come across. But if you overcome such complication, whatever the result what you have got drawn with the animal model, so this can build a chain for the clinical studies and human dose can be get identified by using the animal models. So now let us see about the types of animal models used. So the animal models of the disease can be broadly classified into acute physiological and pharmacological models, chronic physiological and pharmacological models, and the third one is the genetical models. Let us see each type of animal model with some example. Now let us try to understand acute physiological and pharmacological model. As the word acute represents, it's of a shorter duration. So now these models are intended to mimic certain aspects of the clinical disorders. So this can be achieved by giving some of the chemicals, some manipulation we will be doing with the animal. So this can be achieved by changing the physical status, or it may be by giving some of the chemicals. So this can be achieved of so that the animal will show the disease symptoms. So let us consider some of the example to understand how the mimicking aspects will be exhibited in the animals. So now seizures induced by the electrical stimulation, just the manipulation by passing the electrical stimulation in the brain, so that epilepsy will be absorbed in the animal. This for a shorter period. So the one more example, the physical screening technique can be used to check the analgesic activity of central acting analgesics by using a hot plate. This is also an acute model. For a shorter duration, the pain will be absorbed in the animal, how long the animal will be present on the hot plate. So one more example, that is the histamine induced bronchoconstriction as a model for asthma. This is also a shorter duration model. That depends on the dose of histamine what you are given. Once the dose got faded, immediately so animal will get normal, because here you use the normal animal by giving some of the chemicals or some of the physical conditions. We are inducing a shorter duration of disease symptom in an animal in order to check whether the given drug is having action or not. So they comes under acute physiological and pharmacological models. For a shorter duration, the studies can be done. Now the next is the chronic physiological and pharmacological models. So these models involve the use of some drugs or even some of the physical interventions can be done in order to produce some abnormality which are similar to the disease condition. This example, it may be the naloxone to inhibit insulin secretions. So it will act as a model of type 1 diabetes mellitus. Then one more example which can be quoted, that is the self-administration of opioids like nicotine or other drugs as a model of drug dependence. So here for a prolonged period of time, so many times with the naloxone model, it's an irreversible phenomenon where diabetes may turn as a permanent in an animal, but chronically the symptoms would be observed in an animal, and in some condition like chronically the drug administration should be done in order to get the disease symptoms. So this is the difference what you can find in the acute physiological and pharmacological model and chronic physiological and pharmacological model. The manipulation or drug induction of the disease time will be varied in such condition, and the duration of occurrence of the symptoms in the animals also varied. In chronic model, it will be for a longer time. Let us see about the next type, that is the genetic animal models. So now the word transgenic animals, I was telling, like just we are going to change the gene sequences. Sometimes like these transgenic animals can be produced by deletion, or sometimes overexpression of specific genes will be done in order to show some abnormalities that may resemble to the human disease. So the development of these transgenic technologies have allowed inbreed strain to produce with the gene abnormality to produce throughout the animal life. These are advantages. Right from the birth throughout the life, the animal will be having the disease with him. So now earlier two models what you have discussed, acute was for shorter period, whereas chronic model by birth the animal will be healthier, but after manipulation they may get involved with the disease. But in the genetic, this is the difference we can come across with the earlier two models. Like by birth itself they will be getting the disease, and throughout the life the animal will suffer with the disease, so that it is more reliable and suitable model for screening the drug action. So now while doing animal studies, it's essential to validate certain criteria while testing the animal models. So an animal model produced in a lab can never exactly replicate a spontaneous human disease, even though if you are using transgenic animal also slight variations can be observed. So that we will be going with certain validity steps. So this can be done by using three types. So they include like phase validity, construct validity, and predictive type of validity. Let us try to understand what is this phase validity. This phase validity refers to the accuracy with which the model reproduces the phenomenon. Now while using the animal model, what the symptoms they will be showing, so that is called as the phase validity. If the symptom resemble to the pathological changes or characterization of a disease, so then we can use such models, such as the phase validity got passed. Then next is the construct validity. So this refers to the theoretical rationale with which the model is based. That nothing but the extent to which the etiology of the human disease is reflected in the model. While getting a disease, even the etiological reasons should be known. While keeping the animal in such etiological environment, what are the factors they produce the disease, whether such phenomenon have been implemented and whether etiological reason is a reason for disease in the animal, so that will be answered by construct validity by checking the etiological aspects in the human disease. Then the last type, that is the predictive validity. This predictive validity can be defined as a measure of how well a model can be used to predict currently unknown aspect of the disease in human. So now it's nothing but we are going to correlate the therapeutical outcomes from the animal model to the human models. The best example is the use of 6-hydroxydopamine rodent model for Parkinson disease, because 6-hydroxydopamine can induce Parkinson in the animal. This validity refers to the extent to which the effect of manipulation, that is the drug treatment, example we can consider 6-ortho-hydroxydopamine in the model, is a predictive of the effect in the human disorder. So this is the most important of the three, as it is more directly relevant to the issue of predicting therapeutical efficacy. By passing all these three types of validity procedures, such that we can confirm the animal model can have some effect on the drug, or validated models can be